Up first, we have Alex Hudigalong, the Chief Scientific Officer of Levadura Biotechnology. Just as a reminder, again, 5 minutes per presentation, 4 minutes for Q&A. And I think we're just getting everything teed up right now. So Alex, whenever you're ready, I'm not sure if you're unmuted at this point, but it looks like they're getting you pretty well set up here.
There, can you hear me?
Yes, great.
Okay.
All right.
Good afternoon, everyone, for Morgan and the judges for being here, and the attendees, of course. My name is Alex Hutagalang, and today I'll tell you about Levadura Biotechnology, which will be the go-to provider of cannabinoids for the changing cannabis market. Oops. There it is. Uh-oh. I was at the end of my, there it is. Let's start with our team. As CEO with a PhD in biochemistry, I bring the technical expertise of yeast genetic engineering and industrial biotech, while Brian and Matt lead business development, finance, and healthcare, strategy and finance. The three of us have worked together extensively and have taken an industrial biotech process from concept to commercialization. So what are we going to do for the cannabis market? We all know the cannabis market will be huge, and the non-flower market segments, like concentrates and edibles, are expected to be worth billions of dollars in the next 2 years. Both of these markets rely on a consistent and high-quality source of purified cannabinoids. Pharmaceutical applications of cannabis are expected to be worth over $5 billion in the next 7 years as well. Unfortunately, there's a problem. Consistency in chemical composition is one of the greatest challenges to plant-based drug development. Which leads to the question, how will the market efficiently source pure and consistent cannabinoids for these applications? I think we all agree that the cannabis plant is amazing and provides benefits unlike any other plant. However, as a means of cannabinoid production, there's lots of room for improvement. The whole process takes several months, with growing being the rate limiting step. It's a labor, water, and energy intensive process requiring a large footprint, which, if you're outdoors, is subject to the weather and normal agriculture-related environmental issues, as well as all the issues that Chris Booth from TheraCann raised. When it comes to extraction and purification, there are numerous substances that need to be removed: mold, lipids and waxes, pesticides, heavy metals, and aflatoxins, just to start. The end result is an often inconsistent cannabinoid mixture based on the plant and the quality of the crop. Levadura solves all these problems. Our expertise lies in our ability to genetically engineer yeast for a fermentation process. Each engineered yeast will produce one cannabinoid in a 1-week fermentation and purification cycle. Production will require a small footprint and will be a sterile and highly controlled process in fermentation tanks. All this without the known plant-based contaminants. Why is this important? Because Levadura as a means of production can make a big difference to your bottom line. Plant-based production of cannabinoids is handicapped by the time, usually several months, it takes to get product. The only way to increase output is to have better plants or more space. Furthermore, as you will hear from Zach at Polar Extracts today, extraction can be a bottleneck to cannabinoid production. The result, cost of manufacturing for a kilogram of cannabinoid, in this case THC, on the order of thousands. The advantage of a Levadura process is its speed and flexibility. A 1-week production cycle allows for more than 50 cycles a year to ensure flexibility to market demands. A production facility can be located in a small warehouse with enough capacity to produce 50 to 75 kilos of product with each run. And at the end of our fully funded development timeline, we expect to be able to produce a purified cannabinoid at around $350 a kilo. As a technology company, one of our key components is our intellectual property. We have recently filed a US patent to protect the core of our technology, shown below. The key advantage of our approach is how we engineer yeast to convert the input, in our case vegetable oil, to the output, a cannabinoid, and I'll elaborate on that here. Any kind of vegetable oil is composed of triglycerides. Each triglyceride is composed of 1 molecule of glycerol and 3 fatty acids. It's the fatty acid component that is valuable for our technology, which I show you here with the tail portion highlighted in red. By genetically engineering our yeast to control how they consume fatty acids, we have developed proof of concept production of CBGA, THCA, and the cannabinoid precursor olivotolic acid. I've highlighted where the red tail ends up on a mature cannabinoid. I'll tell you more about why that's important on the next slide. By controlling how much of the fatty acid tail we leave behind, we can make rare cannabinoids, such as those of the 4-ol class, which I show you here, with a longer tail shown in green. THCP is found naturally in the plant and has recently been shown to be 33 times more active than THC through binding to the CB1 receptor. Our approach also allows for producing novel cannabinoids. Most plant-based oils contain unsaturated fatty acids, such as oleic, with 1 unsaturation, which is a major component of canola oil, and linoleic, with 2 unsaturations, which is found in sunflower oil. By producing cannabinoids with unusual tails from these fatty acids, the technology would be ideal for pharma applications. To provide some context, I'm showing you the structure of adulamic acid, or lenabasum, in the drug pipeline of Corbus Pharmaceuticals. Notice its unusual tail. It's at phase 2 or phase 3 trials for several diseases whereby inflammation is a critical component of their pathologies. This brings me to our ask.
Yeah, just
based on the progress.
About 5 minutes, okay?
Yep. This brings me to our ask. Based on the progress of our seed round, we are raising $2 million for our Series A, which will be used to produce the following goals: purified CBGA for commercial development, production of CBDA and THCA in fermenters, development of commercial partnerships, and the granting of our US patent, and research to support new patent applications. The pie chart shows use of funds during that time, the bulk of it going to personnel, supplies, and equipment. I'll stop here and take questions.
Great, thank you. Okay, judges, got about 4 minutes, so try to keep it to one a person.
Or should I continue sharing screen?
I'm happy to kick off. Really interesting presentation. I really loved the slide you had regarding extraction versus their process and then the cost implications. What is the CapEx requirements and sort of the licensing requirements to do the process?
So for at the cost of goods of about $350 a kilo, we're talking about our own production facility there, where we would be doing fermentation and extraction all within one facility, and the capex for that kind of a facility would be somewhere around 10 million for those kinds of yields that we discussed in that slide there.
Great. And do you need a cannabis, like a operating license to do so, or a DE?
I believe so. At this stage, I'm not so sure what the licensing needs will be at this stage, because this is a kind of an unusual process within the whole production workflow of producing cannabinoids, which generally applies to the flower and the plant. So now coming from a biosynthetic source, it's not exactly clear to me at this stage what those requirements will be, and hopefully, we'll get some light shed on that as we continue down the process.
Hi, Alex. That was a fantastic presentation. Clearly very interesting stuff. What is the plan and timeline to bringing this to market look like? Based on from where you are, what are the kind of milestones and timeline associated with this?
So I'll just speak about what we're trying to do for our Series A. For that particular raise, we again are focused on making CBGA at quantities sufficient for commercial development, and that just means that we're providing samples to any interested parties that would like to see the quality and nature of our CBGA that we're producing. And the timeline for what we're saying for our Series A is about a year to a year and a half. And at that point in time, it's not clear to me what the market demand is for CBG at this stage. I know it has some traction within the marketplace, but it's not a significant player right now. I have no idea what that's going to look like in a year and a half. But we believe at that point in time, what we are planning to do is go to our next raise. But because we've been able to demonstrate that we are able to make CBGA, we envision a inflection in value, whereby we'll need the additional money to start developing production of CBDA and THCA, which will be, of course, commercially relevant within the marketplace today. And that timeline is about 2 years. So total timeline from including Series A all the way through the development of CBDA and THCA at commercial scale in fermenters, we see a timeline of about 2.5 to 3 years.
Alex,
Having said that, a year and a half from now, I have no idea if CBGA will have any type of significant presence in the marketplace, and if we're commercially, if we're at a commercial production scale or level for CBGA, then there's chances for commercialization there as well.
So it's a good presentation. I've seen a couple companies similar to this trying to get that consistency down, highlighting a couple of things that you said that were spot on. Obviously, you identified the goals and you knew what the investor audience wanted with the IP. You understand the market. What I'd like to know is about needs and roadblocks. You kind of mentioned what sets the business apart, having rare cannabinoids, which is important with consistency and accurate dosing, I would throw in there as well. Added the team and products and created a summary with call to action. But what I want to know is more about what sets you apart and some of the needs and roadblocks that you might face to differentiate you from a competitor.
Right. So there are a number of biosynthetic competitors within this marketplace, which you've highlighted, Josh. What's unusual about our approach is the IP that we're talking about, and that we're going from fatty acids to cannabinoids. And I don't want to dive into the biosynthetic pathway, but there are metabolic advantages in doing that. And by doing that, what we envision is ultimately there's going to be a higher productivity for our process. And in the actual theoretical yield of a cannabinoid from a fatty acid is higher than the traditional route of a biosynthetic approach, which is from sugar. So by having this higher yield, I think we see our advantages are in the productivity of a fermentation, which allows for a 1-week cycle in a typical fermentation, which allows for a significant number of production runs each year, which ultimately is what's going to drive down cost of manufacturing, because it's that productivity or increased productivity, which is one of the significant factors in how much it's going to cost to actually make a cannabinoid within a certain amount of time, based on the input of labor and materials and utilities and capex or depreciation of capex, dividing all those costs by how much product you're able to make in a certain amount of time is really what's going to drive down that cost of manufacturing. So that's what we see as our major advantage moving forward. Great. Having said that, we need the funding to really to make that a reality. So, but that's what we see as our major advantage.